Thank you very much, José Gallardo, first for kind invitation to present shortly about the kind of use of genomics in fisheries and conservation. So as José Gallardo said, I'm coming from Swedish University of Agricultural Sciences, and I'm working with different kind of fish species, but mostly using genomics tools. So today, yeah, as I said, I'll try to introduce you the field and give you some examples about the use of genomics in fisheries and conservation. But by no means it's a very comprehensive kind of overview about it. It's more like to give you a little bit of overview what can genomics do in this context. So. For example, the first question, perhaps to kind of more general question to ask is that what do we mean with the genetics or genomics, and what is the difference between genetics and genomics? Because is that genomics only a kind of a fancy word that people are using nowadays compared to what it was before? And so perhaps, I mean, it is one way true that we are doing lots of genetic analysis with the genomic tools, but if you think about more formally, genetics usually focuses on heredity and kind of a characters that are transmitted from one kind of generation to another, and usually focusing on limited number of genes or loci, while the genomics is the field that really tries to cover the whole genome or transcriptome or all the entirety of the whole organism. And that's called the genome. And how we do it is basically because of the recent developments of high throughput sequencing technologies, we are able to really sequence the whole genomes very quickly and very cheaply, and then generate huge amounts of data that we are using very different bioinformatics tools to kind of make sense of all the huge data that we have collected, and then basically learn something new about different characters of our species or populations. And for those who are maybe not so familiar with the genomes and genomic analysis, the first thing perhaps to remember for you is that genomes are really huge. And just to try to illustrate how big they are, we can assume that, for example, Atlantic salmon genome is 3 billion base pairs long, but that doesn't really maybe give you the kind of really the appreciation how big is 3 billion. So, and then one way to visualize is that if you assume that one nucleotide, one base pair is 1 millimeter long, then one Atlantic salmon genome should kind of go all the way from Santiago to Punta Arenas, where we are now. So, and that's just one genome. And all these information is actually, packed into the single cell, or actually, diploid organisms. There are 2 copies of this kind of this much information packed into one cell, and we are able to now actually to study it as a whole. So I think it's quite fascinating, but also pretty challenging. And then just jumping right away to what genomics can do. So if we are kind of defining that genetic methods usually use tens or hundreds of variable sites of the genome or markers, while genomics tools really study thousands of markers and genes at the same time. And there are different things that genomics can help in, that can help fisheries management and also aquaculture. And as you can see in this slide, there are, for example, areas like evaluating stock connectivity, defining management units, doing something called mixed stock analysis, where you are actually analyzing the kind of determining the origin of fish in a mixed catch, in a way that if one catch consists of many fish from different origin, you can able to actually, with the genetic and genomic tools to separate it, is population size estimates, determining something called adaptive divergence, the regions of the genome that are influenced by selection. You could also use genetics and genomics to traceability and species identification, which means that you can actually, trace the populations of species from the kind of primary fisherman to the product. And very important fields in aquaculture include biosecurity. You can use it, to determine the parentage, like who has been the fathers and mothers and grandmothers and grandfathers of the certain cross. And monitoring pathogens is nowadays a very common place to use molecular and genetic and genomic tools. And you can also use genomics to actually speed up the selection process in aquaculture. So these are very kind of broadly very different themes, and we could spend a lot of time on everything of it. But I thought that within these 20 minutes, I will try to give you a few examples of some of these kind of usabilities and utilities. So I'll jump right into what I'm trying to do today, is basically give you 4 different examples or 4 different areas where genomics can help. And first is identification of species. Second, we go to identification of genetically distinct populations. Then I'll give you an example of how genomics is used to detect hybridization between hatchery and wild fish. And then finally, some words about the traceability of fisheries and aquaculture. So I start with species identification. For example, one nice example is the redfish Sebastes complex. That we know that basically, the redfish is in North Atlantic, a very huge species complex, which means that there are multiple species that look very, very similar to each other, and we are not able to separate them by simply by eye or well, sometimes by morphology, but not very easily distinguish them. But it's a very important fishery stock. So in order to manage it and not to overfish some of the species or populations, we need to know who are there and what are the species and what are the populations in this area. And for example, there's one the study that then sampled redfish along the Canadian coast and all the way to Greenland, and basically then genotyped these individuals by tens of thousands of single nucleotide polymorphisms or variants. And the first figure that you see, the red and the blue bars, means that basically it's the genomic data shows very clearly that there are 2 different species, very distinct species in this sample. So each line here consists of 1 individual fish here. So we could really see that there is like 2 species, which is Sebastes fasciatus and Sebastes mentella. And then the same genomic markers, can be used to further separate different populations, which means that they are genetically somewhat distinct units within the species. And here you can see that these kind of different colors on a figure B. That there, for example, on the right side, there is a dark blue, light blue, and really different colors that indicate that these fish have a very different genetic composition. So it means that we can really, with the genetic markers and genome-wide approach, we can distinguish what are the species if it's not known, and what are the genetically distinct populations in what we are actually fishing. So then I'd like to go a little bit back and think about it, and why it is important to actually understand population genetic structure. And here are the 3 different examples on the right side. You can assume that there is different types of genotypes of fish in the sea, but there is no really spatial structure with that. So in a way that then doesn't really matter where we stock, where we fish, as long as we fish not over the limit that the population still can reproduce and give is in a sustainable level, there's no problem. But the middle case and on the left side, you can see that if the fish in different areas, the fish actually differs to each other genetically. And in case we don't know it, we can very easily overfish or actually, drive some certain populations to extinction, even without knowing it. And then what happens afterwards is that it's not clear that when the fishing stops, the fish stock or population, the same one might never come back and recover because it's kind of fished out. So therefore, we really need to know the kind of the genetic structure and relationship between fish and what we are harvesting. And the next example is about this kind of a very famous kind of, or very widely distributed species, Atlantic herring. And it's one of the world's most important commercial fisheries. And actually, it's one of the most abundant vertebrates of Earth, one of those. And previous genetic approaches using few markers have been not successful to actually separating different stocks. And Atlantic herring has been a kind of a super important, through the human history. And you can see on below there some medieval carving of the herring fishery, for example. So people have been really dependent on herring as a food for a very long time. And here is the one quite recent paper that actually sequenced the genomes of herring from the west and eastern side of the Atlantic Ocean, as well as Baltic Sea and British Isles, and basically sequenced the whole genomes of all of those samples. And what it turns out, based on the huge number, or based on the markers that seem to be under selection, which means that they show very high differences between different populations, we can really separate for the first time these different groups of populations. And even further, we can actually identify different type of ecotypes. Like you can see Baltic Sea spring and autumn, which means that these fish spawn, some of those spawn in the autumn and some of them in spring. And before that, it wasn't known that is there any genetic difference or between them. But with the genomic markers, you can actually very clearly separate those. And furthermore, so genomic analysis really able to identify these loci and genes that are under selection. And here is the example of a plot where, like, every dot in this plot on the left, this is one marker, and these high peaks, figure is also called Manhattan plot, showing the highest differences between spring spawners and autumn spawners. And you can see that actually the same genes are showing extreme differences in the Baltic Sea as also in the Atlantic, which really kind of very nicely demonstrates the kind of parallelism of evolution, that we can see that the same genes are kind of under selection in different geographic locations. And so as a biologist, it's very interesting to go then further to look at each of those genes to understand more what they do and how do they kind of improve the fitness of the organism. And for example, here is one example where one of the genes that was called rhodopsin that actually affects them, the sensitivity of light in the eye of the herring, and how certain mutations change the protein structure and helping kind of the herring to see in these more darker waters in the Baltic. And then on the comparative genomics things, it shows the figure where basically you can see if the same mutations have occurred repeatedly during the evolution. And here is the kind of one example where this kind of a clear water allele is blue, is dominant in a marine organism, and the red kind of red light shift as a red kind of are more common in many species in the freshwater systems. So it's a very exciting to see kind of all the parallelism that occurs in the kind of during the evolution. And what is the relevance for fisheries management for this is that once we can identify those diagnostic markers that are very different between the populations, we can easily separate and use that in a stock assessment, to evaluate what are we fishing. Are we fishing some stocks too much, or are we fishing each stock in the same way, and how are the stocks doing anyway? Like, are the populations overfished or show the decline in abundance or not? So that's really the essence, that once we have the genetic markers to identify populations, we can use it to help the fisheries management. And here is just a random example of, you can estimate, for example, the proportion of each population in a catch in time, that you can see that some populations have been contributing more to the catch to the others, and so on. Yes. And then I'm jumping very quickly forward because not too much time. Another very important area is farm-wild salmon interactions. We know that different in Norway and Chile is growing very much salmonids in the net pens. But what happens is that the net pens get broken now and then, and lots of farmed fish actually escapes, and then if there are wild populations nearby, they also interbreed with the wild individuals, which causes the gene flow from the domestic stocks to the wild. And we know that changes the kind of a genetic structure of the wild populations, and also reduces the population viability in the wild. And it has been this kind of escaped farm salmon, and the introgression has been identified as quite a major threat to wild salmon. And one example of that is that, for example, the Canadian researchers studied both North American and European Atlantic salmon in both sides of the Atlantic. And first, they kind of used genome-wide markers to identify, to ask the question, how different are North American and European Atlantic salmon? And you can see it with the red and blue dots here, that in this kind of imaginary space, two different dimensions, PC1 and PC2, and you can see that basically the blue dots are very, very different from the red ones. So it's like a very deep lineage from millions of years ago. This kind of has evolved. But what has happened now is something that you can see hopefully in this figure, is that if you then compare the wild European stocks in the blue as blue dots, and the Canadian wild stock as red, put onto this the pink ones that are aquaculture fish, you can see that they are kind of getting between the pure European and North American these kind of populations, which means that these individuals have some considerable proportion of European ancestry, and that's also shown on the right side. So it's clear that aquaculture, North American aquaculture salmon, somehow, even it shouldn't have any European residues, they do have quite a lot. And the same actually holds for the escaped farm. So if the North American kind of farm salmon is escaping, here shown as a yellow dot, they also have a kind of this kind of more ancestry of European ancestry, and that means that kind of bad news for wild populations, because sooner or later there will be also these European parts of the European side of the variants turning out in the North America. So to kind of sum up this part is that even we know that European salmon have never approved for farming in Canada. This study actually shows that there is considerable European ancestry, and these kind of genome-wide approaches are very, very powerful to identify these type of genome-wide effects. Yes, and now finally I jump in something called traceability, which means that, I mean, if we are fisheries catch fish, we want to manage the stocks sustainably, and ideally we should know every stage of the kind of where the seafood goes, where it comes from, and what it consists of. And that's where, and also it's also in the fisheries side, and also very important for aquaculture. And that's another huge area where genomics can really help to kind of to fight against these kind of fraud and unregulated fisheries, and really provide kind of the tools to trace your product from the fisherman to the kind of to the plate, basically to the customer. I think I'm already almost over the time. So basically what I would like you to remember from this talk is that basically we know that next generation sequencing and genomic tools have already really transformed the life sciences and biology as we do. And at the same time, we know unfortunately that the vast majority of the world's fish stocks are already, collapsed or very close to the collapse. And so we really need desperately to help the fisheries to drive towards sustainability. And I think that population genomics methods can help towards this goal. It's not the kind of a magic bullet to solve all the problems, but at least it's very much step towards the right direction. So with that, I think stop here and hopefully wait for your comments. So thank you for your attention.
Thank you very much, Dr. Vasemägi, for a very informative and interesting talk. Perfect as an introduction for what we are going to be listening to in the next talks. I don't see any questions yet. I don't know if you received any question directly, but if not, we're going to continue with the rest of the talks, but not before I encourage our participants to feel free to ask any question to any of the speakers at any time. Thanks again, Dr. Vasemägi.
Yeah, thank you very much. So I hope that everything was clear, so no questions.
Yes, that must be it, yeah.
Yeah, bye-bye.
We have a question in the chat
yes.
now.
Daniela is asking us what the terminal effects are expected to occur from introgression into wild salmon.
Yes, that's a super nice question and very interesting one, because there are different, the effects depend on the environment, but generally what has been seen is that, for example, the farmed salmon, for example, the farmed salmon, basically it grows much faster, but also has a much lower survival in the wild. So, which means that if the farmed salmon introgresses with the wild ones, they also have very different life history traits. They grow fast and they might not mature very early, and they have very different behavior. And at the end of the day, it's not good news for population kind of sustainability and survival. So maybe that's the kind of a very simple answer.